All right, welcome back to the 2025 Borealis Psychedelic Science Summit in Stockholm. My name is Yoshi, and I'm joined now by Dr. Bálasz Szigeti.
Thank you for having me.
Welcome. How are you doing?
Tired, but good. A conference like this is intense, but it's in a good way.
Yeah, you've been here the full 2 days?
Yes.
Right. Seen a lot of presentations. Had some presentations?
Correct. Yeah, I'm just coming from mine, so it's out of the way, so now the party can start.
So now you're done with all of yours.
Yeah, I mean, I still got this podcast, but after that, yes, I'm done.
but I'm nice. I'm not.
Go easy with me.
This will be gentle. Yes. Tell me a little bit about your background and how you got to working in the psychedelic field.
So I actually studied physics and then did a PhD in computational neuroscience. I was working on the behavioral classification of C. elegans, which is this bacteria-eating worm that lives in the soil. And then, it's a fairly typical story that I started to go to raves and had some personal experiences that made me want to join this field as a researcher. Yeah. So I was already in academia, but I was doing something not related to psychedelics. But at some point, I just realized that I'm much more interested in this than what I was doing back then. Yeah. So I made the switch, which was a difficult transition. In academia, typically you're logged into a topic and then that's what you do. So it's been a hard transition. But anyway, here I am now.
Nice. So how did you transition, or to what did you transition in the psychedelic field?
So for me, how I became, when I was interested in the psychedelic science, I realized that I need to, do something splashy to make a name for myself. And back in 2017, microdosing was starting to get hot. And back then, there were no placebo-controlled studies about microdosing. And I tried to raise funds for one, but I failed miserably, not being a medical researcher. And in that frustration, I realized later that actually you can do placebo-controlled studies outside of clinical studies. And this was the start of today what is known as the self-blinding microdose study. So I worked out a method how people at home can implement their own placebo control without clinical supervision. And the gist of the method is that you hide your microdoses inside non-transparent gel capsules. Empty capsules can be used as placebos. Then you label with QR codes and you mix this together. And when you scan the QR code that link with the IT infrastructure of the system, long story short, at the end of the day, you threw away half of your capsules, kept the other half, but you did not know whether you have the placebo or the microdose capsules. And then I was having a job in New York when I was coming up with this idea. And at some point, I realized that I can't have my daytime job and do this study. And this was the time when my second daughter was born. So as you imagine, it was a pretty difficult conversation with my wife and my parents that I'm going to give up my cozy job in New York, move back home without any salary or institutional support, and do this self-blinding microdose study on LSD with the methodology that nobody has ever used before. But I believed in it so much that I did it nonetheless. And this study was published. Now it was published in 2021, and it is still the largest placebo-controlled study on microdosing. So that's how I got into psychedelic science. I did this study, and it's a study which is it's well known in the psychedelic space.
That is amazing. I have so many questions. First one is, how did you even think of to make a splash? What made you think of that? I mean, working from, was it bacteria eating worms?
Yeah.
It's not the splashiest of worlds, studying physics and studying that kind of worms. And then you realize, I have to do something splashy to make a name for myself.
because I wanted to, transition into psychedelic science. And it is already back then in 2017, it was a very saturated field. It's an exciting field. A lot of people want to come into that. So if I would have reached out that, hey, I want to be in psychedelic science, nobody would have answered the call probably. That's just how it works in academia, So, and especially because I was already doing postdoctoral work in another field, I just knew that I have to put myself on the map somehow. And I was wrestling with this, and this idea for the self-blinding microdose study came, and I believed in it and, pursued it. And of course, this was not an easy study to do.
Yeah.
Imagine when we applied for the IRB, for the ethics approval, that hey, people are going to do drugs at home and they're going to do their own placebo. It was a hard sell. But, in the end, it worked out.
Where did the idea come from?
I was reading a book by Irving Kirsch. It's called The Emperor New Drugs, which is about placebo effects in antidepressant treatment. So I was thinking about these ideas, and actually the idea came on a sleepless night. This when I had a small baby at home, and I was actually burping my daughter in the middle of the night when the idea hit me. And I was so excited that I didn't sleep the entire night. And in the morning, when my wife woke up, she's like, how was the night? And I was like, I haven't slept. I have a pretty good idea, but it's going to be a lot of work to implement it.
Yeah.
So, as soon as I had the idea, I knew that I'm onto something. She can verify the story that really in the morning I said it is that I get a good idea, but it's going to be a lot of work.
That is so interesting. Many people have what they think are good ideas during sleepless nights, but they don't usually turn out this good.
Don't worry, I had plenty of those as well, but this is the few exceptions that did work out.
It's a very, very good exception. All right, so what has been your journey since the study was published? What have you been doing since, and what are you currently doing?
So I am a little bit lost faith in microdosing. I initially wanted to do that study also because I was having positive personal experiences with microdosing. And I often joke that I am one of the few researchers who I hate my own research. I really do. I consistently design studies to show something, and consistently I find the opposite of that.
Oh, wow. So,
for example, the motivation to do this self-blinding study, of course, was to show that microdosing is not just a placebo effect, but of course what we find is that the improvements in the placebo arms are equal to the improvements in the microdosing arms. So, imagine that, I just told you that, I gave up quite a lot, to do this study. I was really believing in microdosing, and then to get this result, it was quite disheartening.
Yeah.
And especially because after this, I became a little bit of an anti-hero in the psychedelic community. Yeah. I wanted to be the cool guy, who shows that it's not just the placebo effect. And hey, actually, guys, I'm sorry to say, but it might be just a placebo effect. It was, it's a weird mental state. And, And anyway, so I lost faith in microdosing, but I still remained very interested in these questions around functional unblinding. That's the technical term when patients can figure out their treatment allocation in a study. So typically you do placebo-controlled studies in a blinded manner, which means that you don't tell the patient whether they are receiving placebo or the active treatment. But with psychedelics, very often they can figure it out based on the strong subjective effects of these drugs. And this introduces all sorts of biases into the interpretation of these studies, and I find this topic very interesting. And right now, a lot of what I have learned in trying to do this placebo control. Sorry, I'm gonna backtrack here a little bit. So with the microdose study, what we figured out is that, even with microdosing, patients can figure out their treatment allocation, so whether they take the microdose or a placebo at a better than chance rate. Mm-hmm. So, if patients can do that in a microdosing study, then of course those biases are going to be much more significant in the large dose studies.
Right.
So, then, sort of lost a little faith in microdosing, and right now I'm trying to apply the same methods for large dose studies, but because they're it is even that much more obvious, it's very challenging to figure out what is a placebo effect and what is not in these psychedelic studies.
Right. So how do you do that? How do you?
The short answer is that you can't really do it. Yeah. There is just there's a lot of people have tried methods like active placebos,
like caffeine. I think I've heard.
Yeah, and niacin is a popular one, but it's kind of stupid. Anybody who's ever taken psychedelics know that nobody will confuse niacin with psychedelics, right? Yeah. It is just very obvious. And in particular, psychedelics have very characteristic effect. For example, the visual effect. There's no other drug that would give you something like that visual effect. So I somewhat gave up on this idea of blinding, and there are two reasons for that. One of them is this practical reason that you cannot really do it. The second one is that the reason we are running these studies is to isolate what researchers call the non-specific effects from the specific effects. So the non-specific effects are basically, like for example, the placebo effect, it's non-specific. It's not specific to the treatment. Every treatment contains it.
Right.
But what's important to notice here is that, like, when patients undergo a medical treatment, they are also getting these non-specific treatment effects. Like, in research, we go at length to try to isolate what are the specific effects of the treatment. But in every real-world situation, like, the patient is also getting the non-specific effects. So, like, we go at great length, to try to isolate the two. But if we care about like the patient improvement, actually we should embrace these non-specific effects. And rather than try to extract them from the data, we should try to amplify them. At the end of the day, in my experience, patients don't really care about whether they get better because of the placebo effect or non-specific effects. They care about whether they do get better or not. Patients are not going to complain if they get better from placebo.
What are there other non-specific effects apart from placebo, or?
Yeah, so the Hawthorne effect, which is like the effect that you are being observed, or regression to the mean also comes to mind. So like typically patients enroll into these studies when there was a worsening of their condition. Like you have some chronic back pain and it's bad. Suddenly it's really, really bad. That's when you go and sign up for the trial. But then typically, like, from it very, very bad, you just go back to normal bad in a week or two just on its own.
Okay.
There's just this natural fluctuation in the disease's trajectory.
Interesting. Yeah. All right. So your current work is more along those lines, to not separate non-specific from specific, or?
I mean, so what I do right now a lot, what I talk about now is what I like to call the nocebo effect in psychedelic studies. And what that is that it turns out that in psychedelic studies, there is a very large missing chunk of the placebo effect. So if you're looking at traditional antidepressants, SSRIs and SNRIs, then on the Hamilton Depression Rating Scale, which is a typical measurement of depression, the placebo response is about 8 points. It's fairly universal. Like, we have run hundreds, if not thousands of studies with SSRIs. The placebo response is 8 points as measured on this Hamilton Depression Rating Scale. And it's fairly consistent. In psychedelic studies, it's 4 points. So it is like half as much, which means that there is like this weird placebo suppression effect going on. And what I think is happening, and this is just based on informal conversation with some of our patients at UCSF and like, just with the therapists as well, is like, this is disappointment knowing that you have been randomized into the control condition. Like, enrolling in these studies takes quite a lot of effort. You have to complete all of the stupid questionnaires that me and the other researchers are asking you. It's quite a bit of effort. And then, particularly in like psychedelic assisted therapy, like you spend hours of therapy being prepared for a transformative, spiritual, life-altering journey. You talk and talk and talk about it, and finally your dosing day arrives and your spouse drives you to the medical center, and then they are doing like they put on some meditation music, and you finally take the pill and nothing happens. I would be disappointed for sure.
Yes.
And the reason why this is important is that so in medical research, like typically what we are focusing on what is called this between arm effect. And that is or another way of saying that's the treatment versus control difference. How much better patients in the treatment arm did relative to the control. But like, I just told you that there is a missing placebo response in psychedelic studies. So it means that like, you get this treatment versus control difference gets magnified because there is a much lower effect in the control arm. So like, as I told you, in traditional antidepressant studies, the placebo response is typically 8 points. And then the treatment versus control difference is 2 to 3 points. So it adds up to about 11 points. That's the patient improvement. The total patient improvement is the sum of these non-specific effects, which we measure in the placebo control condition, and then the between arm difference, 8 plus 3, 11. In psychedelic studies, the between-arm difference is 6 to 7 points, again on the aforementioned Hamilton Depression Scale. And when you compare the 2 to 3 point improvement in the SSRIs and you put next to it the psychedelic 6 to 7, and what they're saying is that, look, psychedelics are so much better. But that's not the case. Psychedelics have this large between-arm effect because the placebo response is much weaker, lower, yes. So like, you know, in psychedelic studies, the placebo response is about 4 points, and then the between-arm effect is about 7 points. 4 plus 7, also 11. The patient improvement is actually the same between psychedelic studies and SSRIs. But the traditional way that we are looking at these trials is just this metric of this between-arm difference. We are only looking at it relative to the control. But it turns out that with psychedelics, this control condition has a very low effect size. Right. And that introduces this. If you're looking at just the between treatment effect, you are assuming that the control condition did roughly equally good between these two studies. Right. But it turns out that no, in psychedelics, there is this very significant and very unique placebo suppression effects that biases the results.
So interesting. So in the end, what do you do with that information? Do you have to, you mentioned nocebo, do you have to do trials differently without placebo or?
I mean, you know, that's kind of like, you know, what everybody is trying to figure out. And I think, you know, where the field is that there is like, you know, people are talking about how to improve the situation. And I think there are ways to improve it. Like, for example, something that's called like modified informed consent, when you don't tell, for example, the number of arms in the study. Like you tell them that everybody is going to get psilocybin and you use 1 milligram as a control condition. And these things can improve blinding, but I don't see how we could get to like an actually blinded psychedelic study. And bear in mind that this issue is not just for psychedelics. Like, you know, you can't really blind meditation interventions either. Right. So to me, the situation is weird because the whole how we evaluate interventions is based on these double-blind studies. Like that's the paradigm that we use. But if you think about like the space of all possible interventions, most of those interventions are not blindable. Behavioral interventions are not blindable. Like, to stick with the example of meditation, a blinded meditation study would need to involve patients meditating, not being aware that they are meditating. Right. That's difficult to do.
Very difficult.
So there is this big space. Most of the interventions are not blindable, even in theory. Then there is a smaller space, pharmacological interventions, where in theory it is blindable in the sense that, hey, I either give you the active pill or the placebo pill. But it turns out that even within pharmacological interventions, due to side effects, even there, like, blinding is extremely difficult to achieve. So, like, you know, we have like this tiny, tiny section of all interventions where blinding actually works, yet the entire framework of how we are evaluating drugs pretends that like this thing, this small intersection, the only thing that, like, you know, if everything would be like that. Right. So I think, you know, it's just a weird situation where the regulators are wanting to do something, which at the end of the day is pretty stupid. Yeah, definitely. I've never thought of it that way.
So is there talk about changing the whole structure to value different types of trials equally as double blind?
So, like, you know, right now there is kind of like, you know, the global brain trust of psychedelic study design is here at this conference. And, you know, when I talk to colleagues about it, I think, you know, many of them would many of them have similar sentiment as just what I expressed. Yeah. That does not mean that regulators see the same way. And I mean, like, you know, how bureaucracy works, there is a lot of politics to it. And like, you know, these just tend to be like very high inertia systems. Like, you know, changing anything takes a lot of will and a lot of time. If you would talk to the FDA, I think there would be a lot of people there who would agree with what I just said. But then, you know, for an agency to like completely change how we evaluate drugs, it's just kind of like, you know, it's a big chunk. Yeah, it's a big ship to turn around. And it is not, and you know, it is, you know, regulation, it's not a scientific question. Like, I think, you know, most people would agree on the scientific points, but, you know, how we translate it into changing the regulation, that's much more politics and policy. Like, you know, that's yeah, completely different matter. Yeah. Okay, so you've done the groundwork to show why this is hard, and people have tried to find a solution, and we're realizing that we somehow need to change how we regulate these
these trials, I guess. Is that am I interpreting that correctly? We're at that stage where, all right, we've tried all the different variations. We need to do something different with regulations for these.
I think, most people in the field would agree with that. And this issue of blinding, most psychiatric medications suffer from that. Like, what has been the strategy of psychiatry is just not to tell patients whether they're getting the treatment and the placebo, and just pretend that patients actually do not know. Basically the field has put its head in the sand, avoiding this problem. But with psychedelics, the problem is so obvious that you can no longer ignore it. And here I want to be fair. If you take, for example, traditional antidepressants, I think there is also some functional unblinding with SSRI trials, but not nearly as much as with psychedelics. So if you have a trial with 2 arms, the way that you can measure blinding integrity is to ask patients to take a guess whether they have received the placebo or the active treatment at the end of the study. And in a 2-arm trial, if it's truly blind, they would guess correctly about 50% of the time. That's just, getting it right by chance, right?
Right.
In psychedelic studies, this correct guess rate is somewhere between 90 to 95%.
Yeah.
With SSRIs, it's about 60%.
Okay.
So, blinding is not perfect with SSRIs either, but the magnitude of the problem is much, much larger with psychedelics. But some people also get it wrong with psychedelics. And one day, I really, really want to do a study of the people who cannot distinguish 200 micrograms of LSD from placebo. I'm just like, what happened?
Who are these 5 to 10%? What's going on?
Yeah, exactly.
That would be very interesting to find out. Wow. All right. Well, switching gears a little bit, tell me about, in the broader field of psychedelics, psychedelic research, what's something you're excited about? What's happening now in the field that you would like to highlight?
I'm going to be biased here, and I'm going to say that right now at UCSF, we are running a study called PDP2. It's a psilocybin for Parkinson's disease. And the reason why that's interesting is because it's not a mental health condition, Parkinson's disease. It's a neurodegenerative disease. So it is just a completely new application of psychedelics. And I know that people are also doing LSD for Alzheimer's. So, this is going beyond those traditional application of psychedelics in the field of mental health. Now we are expanding how psychedelics can be used. And I think that's just exciting. To be honest, one more psilocybin for depression trial, it's a little bit been there, done that, This is the next frontier, conditions other than mental health conditions. And, psilocybin for Parkinson's, we did a pilot study. So it's your typical, small sample, open label study. But what was interesting there is that not only the depression scores improved of the patients, but also their symptoms associated with Parkinson's. So tremors, for example, and their motor functions have improved. So that's why, I'm very hopeful that this psilocybin for Parkinson's study is going to turn out positive results.
That's very interesting. We've talked a lot during this podcast about the mental health aspects, of course, and it's kind of easy to understand why psilocybin or LSD could be helpful in that area. What's the mechanism behind it possibly being helpful in Parkinson's? That may not be as obvious to people.
I mean, it is a neurodegenerative disease. So, there is some sort of a destruction of the nervous system. On the other hand, psychedelics have shown to induce synaptic plasticity. So, they reinvigorate the brain's ability to grow new connections between neurons. So, it could counteract that neurodegenerative processes that are taking place. So, there are no mechanistic studies on, how it could be useful in Parkinson's, but, the big that's how you can put together the big picture, neurodegeneration, and you can counteract that with the synaptic, with the new synaptic plasticity induced by psychedelics potentially.
That is so interesting. Since there's a neurogeneric, is that the word? Effects?
Neurodegenerative.
Yes, but from the psychedelics, they are generic, right?
You can say that. Yes. Okay. Yeah.
Since they might have neurogeneric generative. Thank you. That's the word I'm looking for. Neurogenerative effects. Are we looking, or is someone looking at prophylactic use before getting Alzheimer's or Parkinson's, if, like, I don't know, microdosing psilocybin or something like that to prevent people from getting?
No, like generally, like, you know, in medicine, we don't really do preventive medicine trials, like, which is funny because like in public health system, there is a big emphasis on prevention. Like it's much easier to prevent things happening than treating. But from a trial perspective, like, you know, it's already difficult to enroll enough patients in a trial. And like, you know, right now you would really need a super large sample because most of those people are not even going to get the disease, right? So like, you know, it is... Running trials on preventative medicine is something that sounds really good in theory and we should be doing it, but in practice it's a nightmare. And like there are some trials, but it's a very small fraction of the whole biomedical research enterprise.
Could it be done with the method that we began talking about, the one you figured out?
Yeah, the issue with these sort of things is that, you know, for these preventative medicine studies is that you need to keep the patients engaged for a very long time. Like, you know, even just engaging patients for a few months, that's already difficult. Like, you know, it's, you know, the loss of follow-up, like, you know, that's the technical phrase when, you know, you're reaching with the patient who you treated who does not respond. Like, it's already significant, like 3 to 6 months out. For a preventative medicine trial, for something like, let's say, like Parkinson's, like, you know, you would probably need to track people for years. And it only can happen if there are like very strong incentives. Like maybe there's a financial incentive to like respond to every questionnaire, but it's going to cost you a lot of money.
It's a tricky one to figure out. This has been super interesting. Thank you so much for sharing all of this. Where can people find and follow your work?
I mean, like, you know, LinkedIn and Twitter. So like, you know, the usual ones. I'm Bálasz on Twitter, and on LinkedIn, if you put in my name, it's going to come up as well.
Fantastic. Thank you so much for sitting down and having a chat with me.
Thank you for having me.
Much appreciated. And thanks to everyone for tuning in. My name is Yoshi from the Borealis Psychedelic Science Summit in Stockholm. Catch you next time.